Okay, let us move to the next section. It's connective tissue. Okay, so this is basal lamina. We'll just go deeper. So this purple environment will be the connective tissues. Basically packed with many insoluble fiber here. Okay, and the key ECN components in connective tissue will be the fibrillary collagen, also collagen. Okay. And the glycosaminoglycan GH is kind of polysaccharide, and the proteoglycan is the protein conjugate with different sugars, and the multiple adhesive protein and elastic. And those ECN components are produced largely by the fibroblasts, largely by cell. Okay, basically those ECN are super huge. They are even larger than cell. How can cell to generate a component larger than self? That would be weird. And also, it's basically synthesized. They are protein. They are synthesized by secretory pathway. As you can see here, based on electron microscopy, you will see the cytosol is pretty dark. It means the cytosol is super crowded. People have measured the pore of the cytosol. Actually, the protein or molecule larger than 20 nanometer, they cannot freely diffuse in the cytosol. Once you put the molecule larger than 20 nanometer, they were stuck in the cytosol. They cannot move anymore. It just keep accumulating inside the cell. Okay, so to deliver the big molecule, that is why cell need to use vesicle. Okay, and to deliver and put the cargo inside the vesicle. And the average size or the diameter of the vesicle is about 30 to 60 nanometer. Then they will bring this put the large cargo in the vesicle and use a motor protein to drag those vesicle in the cytosol, because the pore of the cytosol is 20 nanometer. So you need to move the component larger than 20 nanometer. That is why to move the vesicle you need energy. That's why the motor protein need to use ATP hydrolysis to move the vesicle toward the destination. That's the way cell to deliver the large molecules. Okay. Okay. So but in the cytosol, it's crowded. So ideally you just cannot synthesize this during this protein synthesis of those ECN. Don't put those protein to the cytosol. Otherwise, they cannot go to any other place. Okay, the one place inside the cell would be not so crowded. It's the lumen of the different organelle. Okay, like an ER lumen or Golgi lumen, they are pretty kind of free space. They can put a lot of different things there. Okay, large cargo here. So it's also the case for the collagen synthesis. So this will be the ER lumens. So once the collagen mRNA synthesize transcript translation and then go out from the nucleus, they approach to the ribosome. Ribosome receive those collagen mRNA. They will approach to the ER. Then they become the ROF ER. Okay, and they synthesize the collagen polypeptide. Put that to the lumen because it's not a crowded environment. Okay, outside in the cytosol is super crowded. You don't put the polypeptide or such a large protein to the cytosol site. So you put in the ER lumens. And those protein, they will use, they will kind of form the disulfide bond to assemble the three polypeptide together. And through those kind of side chain, they will form the zip-like, rend up and form the zip-like patterns, and form the globular domain in the N-terminal and the C-terminal. And they kind of move to the Golgi lumens. In some case, if those protein larger than vesicle, they cannot deliver through the vesicle. They just directly transfer from ER to the Golgi. Okay, the lumen of the ER and the lumen of the Golgi are huge. So they just directly exchange such an environment and then directly deliver those huge cargo to the next organelle. In some case, the Golgi can also directly contact with the plasma membrane. So after modification, they can directly release, by doing this, they just connect with the plasma membrane and release the huge cargo to the extracellular space. They don't need any vesicle, okay? it's pretty similar like this situation. So the pro-collagen, so several chaperone protein, like HeSH protein 47, because during the protein synthesis, they haven't folded to the proper kind of 3D structure. They were exposed to some kind of domain or motif. In this case, in some cases, they will enter with other unnecessary interaction. So the chaperone protein will bind to this site to prevent such a immature protein interaction. That's the function contribute from the HeSH protein. Okay, so HeSH protein 47 will help them to stabilize such a protein structure and prevent immature or unnecessary protein aggregations. Then they will use their kind of domain to do the lateral association to light up together. And finally, we release out to the extracellular space. Then they will cut off those kind of the globular domain. Then this structure can act as a building block. They will assemble together and finally form a huge collagen fibril. So the length of this is about 67 nanometer. It's pretty similar to kind of maxima size that the cell can deal with. Okay, yeah, that's the way. And finally, form such a collagen, they can form a super huge collagen fiber, even larger than a couple thousand nanometer. Okay, so usually this diameter of the one mammalian cell is like a 10 to 20 micrometer. Okay, so the collagen could be the larger because they synthesize in the cell, they synthesize the building block. After releasing to the extracellular space, they assemble outside the cell. Okay. So type 1 collagen, they will enter with a type 6 collagen, and they can form the thicker collagen fiber. And the type 2 collagen, they will enter with a type 9 collagen, and you see this angle here, and it will enter with other type 2 collagen. So finally, this one will form the thick collagen fibril. This one can form the meshwork because it enters with other collagen. Okay, so you can find that is why you will usually find the type 1 collagens. They are all orientated in the same direction, and you will find a lot of type 1 collagen in the tendon because the force will apply in the same direction. So the orientation of those type 1 collagen will all arrange in follow the force that apply to this direction. But in the connective tissue or cartilage, the force could come from the different direction. So it usually will composed by type 2 collagen. Okay, you only need the meshwork. Okay. And this will be the GAG, is a glycosaminoglycan. Okay, so GAG, they are long linear polymer of the repeating disaccharide. Disaccharide is a 2 sugar. Okay, disaccharide. So there will be several the major type of the disaccharide is hyaluronic, heparin sulfate, chondroitin sulfate, and the keratin sulfate. Okay, they all kind of 2 sugars and contain many negative charge. So these guys will enter with many waters. Okay, you can imagine that they will enter with many waters. Okay, and if you take a look how those sugar conjugate with the amino acid. So they will only conjugate those sugar can only conjugate with the 3 kind of amino acid: serine, surrounding, and asparagine. Okay, and they will use these 3 linkage sugar to connect with those 3 amino acids. If they link to the serine and the surrounding, they will enter with such an OH group. So we call this O-link oligosaccharide. If they link to the NH2 groups, they will call the N-link oligosaccharide. Okay, yeah. And then after this linkage sugar, then you will have a kind of repeated disaccharide. Those are GAG. Okay. And those GAG, they are really important for several functions. For example, will be the endothelial cell of the first layer of the blood vessel wall. Okay, and this will be the blood vessel lumens. So on the surface of the endothelial cell, there's several surface protein or the sugar like or the GAG like a heparin sulfate. It's a detailed structure look like this. There are several sulfate groups will can bind with the AT3, which stands for the anti-thrombin 3. Okay, this will be the inhibitor of the thrombin. Thrombin will be the protein to induce blood clotting. So if you activate anti-thrombin, it will inhibit thrombin, so prevent the blood clotting. Okay, if you don't have enough amount of heparin sulfate, you will trigger the blood clotting. Okay, that will be the function. And this will be the hyaluronic structure. So basically, the hyaluronic molecules is only here, the yellow one, okay, the right here. So it's also called hyaluronic acid, HA. It's the HA mainly synthesized from the plasma membrane. So this will be the protein hyaluronic acid synthase. On the plasma membrane will synthesize HA and directly release to the extracellular space. Then this hyaluronic molecule will also conjugate with several connecting protein, for example, aggregates, and many linkage sugar will bind to those aggregate protein, then come with a different repeated GAG with different lengths. Okay, and finally they can form such a huge GAG complex. And you can imagine they are negative charge. They will be bind with many different waters. It will generate kind of gel-like environment there. So the cell around here, in some cases, the cell need to have some space to do their proper or more complicated job. For example, they need to proliferate and they need to divide. They need to migrate to the other side. If the environment too compact, they cannot do these things. Okay, so they need to kind of especially in some migrating cell, they were covered by a lot of hyaluronic acid on the surface of the cell. It will help them to kind of generate more space, and they can help them to do the migration in the human body. Okay. Okay. So in the connective tissue, fibronectin, again, it contains a lot of binding site, and also it will be a really important binding site RGD, allowing the integrin to bind with. So this will be the RGD sequence allowing kind of integrin to bind with those of fibronectin. So this will be the kind of experiment researcher previously conduct to prove RGD is really important. So they synthesize different amino acid polypeptide. So it's a D, RED, SRC, G, KGD, RGE, RAD, RGD, blah, 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 RGD, blah, blah, RGD, RGD. And they code those polypeptide on the culture dish with an increasing concentration, then test how many cells can adhere to that culture dish. And then finally found only polypeptide contain RGD can really improve the cell adhesion. With other combination cannot. Okay? Yeah, so the integrin only recognize anything containing RGD. Okay? So this will be the elastic fibers. It's kind of can be found in the connective tissue. There will be the elastin to form such elastic fibers. So this will be the even thicker than collagen fibers, as you can see here. Okay, they also can kind of support the mechanical, give a mechanical support to the connective tissues. Okay. As I mentioned before, when cells need more space, for example, they need to proliferate and they need to do even apoptosis, or they need to do morphogenesis, they need that kind of more space. Okay, to compact, if there's too many extracellular matrices do not give them enough space to do this. Okay, so they can, in this case, they was kind of produce several enzymes like NMPS, okay, metrometalloprotease. They kind of enzyme can digest the protein. They are the major guys to digest extracellular matrix. So those NMP will digest the extracellular matrix around them and generate more space. So AMP, ADAM, or ADAMTS are the enzyme to digest the ECN. And of course, this reaction needs to be tightly controlled, need to be precisely controlled, otherwise there will be the disaster, right? So they have a break. It's TIMS or RAC they will be the inhibitor of those enzymes, so they can be even stop the reaction or inhibit those reaction. Okay. So in this section, we just talk about how cell synthesize the huge collagens. They basically synthesize through the secretory pathway, but they will keep them will do this job in the lumen of the organelle, which can give you the more space. It's not so crowded for them to synthesize a huge protein. And we also introduce about the GAG and the proteoglycan and the O-link and the N-link oligosaccharide. And of course, type 1 and type 2 collagen, they can form they can form the fiber and mesh-like network. And you can find that in the tendon or cartilage, okay? And also the two examples of the GAG function, the heparin will bind with the anti-thrombin 3 and inhibit the thrombin. And the hyaluronic acid, they will contain a lot of, bind with a lot of water and generate gel-like structure, okay? And multi-adhesive protein, they will contain RGD domain and can allow the integrin to bind with. Okay. And also introduce really quickly about elastin. And three family of the metalloprotease, MPS, ADANs, and ADANTS, they kind of can remodel and degrade the ECM for them to do more activity that need the kind of space. Okay. So in the previous section, we only talk about static situation. The cell just there, okay, they don't move, but inside our human body, those are dynamic environment. So such a kind of cell-cell adhesion or cell matrix adhesion is also important for the dynamic process. For example, during the EMT, once they lost the adherent junction or those are tight junction, they will become the mesenchymal like a cell. They need to migrate, but like our working, we also need to deal with the floor, right? They also need to attach with the extracellular matrix to conduct their cell migration. If you carefully check the culture cell, they adhere on the culture dish, they are not totally lie down the bottom. They actually stand up on the culture dish, okay, through those we call the adhesion sites, okay. So if you label the component of those adhesion sites, you will see this is one cell. Those are green signal, they are the adhesion site. Okay, so they use this to stand up there. And during the cell migration, this is a side view of the migrating cell, and they will generate new adhesion site. And from the rear side, they will remove the adhesion site. So such a process needs to be kind of really dynamic, like what we are working. We need to make a new step and remove our the food. Okay. Okay, and inside the adhesion side, you will see many integrin here. It will contact with the extracellular matrix and connect with the other adapter proteins. So that will be the live cell. You will see the green signal is a focal adhesion complex. You will see the cell migrate, and you will form the new focal adhesion complex there toward their destination. This will be also, it could be the really dynamic. If like not so dynamic, cell cannot migrate. If you knock out a certain essential molecule, they become not static, the cell cannot migrate. Okay? So you will see such a form the new adhesion and remove the old adhesion need to be kind of really quickly turned over. Okay? And such a reaction can be controlled by changing the integrin activity. Because I mentioned before, the integrin will be the major one to help cell to adhere to the bottom or to interact with the ECN. So several ways can regulate the integrin activity. First is regulate integrin expression level, but it's too slow. As I mentioned before, if you want to kind of regulate the integrin based on the protein expression level, the process will be super slow because once you receive that signal, you start to express the certain proteins. Take about 12 to 15 minutes. So if you want to accumulate enough amount of the functional integrin to make it any different, it could be the couple hours process. Okay, but the migration should be faster than that. So we need a faster regulation. So protein already there. it's not mainly through the expression regulation. It's through the conformation change, like the gap junction. If you want to control the gap junction function by expression level, it's too slow. You will kind of make change through under the scale about hours. Okay, that's too slow. But if you through such a modification to change their conformation like a phosphorylase, it could be the millisecond scale. Okay, it's also the case here. So the integrin, if you bind with a certain, their functional site will be here. That will be the functional site to interact for the integrin to interact with the extracellular matrix. If you bind with a certain cytosol molecule here, they will make them change their conformation and make them kind of really straight and exposed to their kind of active site to interact with ECN. Okay, without those activating signaling molecules, they will bend it and hide their kind of functional site, so they don't interact with the ECN. So in that will be the inactive conformation. So that will be the protein structure of inactive conformation. So under electron microscopy, you will see, because those are integrin, they are membrane protein, and you need to generate certain kind of membrane microenvironment for them to stand out in your sample dish. Okay, so here they use so-called nanodisk. This will be kind of collect many phospholipids here, and as you can see here, they will be kind of in the aqueous solution. This side will be really kind of hydrophobic layer will be really kind of exposed to the water solution, right? So they just use the alpha helix to rend up and cover this hydrophobic surface interface and hold this system very stable. So they can make a tiny disk to mimic the membrane structure and purify the integrin protein and insert here. So we see this is a nanodisk, and this will be the integrin, and this will be banded structure, and this will be kind of stand-up structure. So to form the new adhesion molecule, those cytosol signal will bind there, and they activate the integrin, and they form the new adhesion. In the rear side, they will kind of remove those activating signaling and the banded integrin. That is why they will remove the old focal adhesion complex. Okay, so platelet will be kind of really important to activate the integrin to interact with others to form the blood clotting. Because we don't have enough time, so I will kind of skip this video. So basically, I just used this cartoon to show you the idea. Okay, so when blood vessel damage happens, this is the blood vessel, and the first layer will be the endothelial cell, then basal lamina. Basal lamina contain many collagen here. When accident happens, the blood vessel damage happens, it will become kind of collagen will expose to the blood vessel, right? And once those platelets touch the exposed collagen, will activate those platelets and trigger the downstream signaling. So the integrin from the platelet will be activated and change their cell fate. They will generate more cytokine or generate more signaling molecules, and they will also activate integrin. So those are integrin from the one platelet will interact with other active neighbor platelet, and through those fibronectin, finally will accumulate a lot of platelet here in the damaged site or in the damaged site. That is why they will prevent the blood leakage. Okay, that will be kind of to trigger the blood clotting through the integrin interaction. And also IgChem, which we showed you before in the beginning. We showed there's a 4 type of the chem here. So the IgChem contain many Ig domain, which is a bowl-like structure. So Ig stands for immunoglobulin. It's originally identified in the antibody, and finally people realize there's many Ig domain can be found in many different proteins, not only antibody. Okay, so this domain contains 70 to 110 residue, and IgChem, their function do not need the calcium. Cadherin need the calcium. IgChem do not need the calcium. If those can mainly function in the neuron cell, they will call the neuronal camped or uncamped. Or intercellular camp will be the ICAM, they function in the movement of the leukocyte into the tissue that we will introduce more. And the GEMs will be the one actually we already introduced before in the tight junction. They are the major camp of the tight junction. Okay, so here we will use an example to show you how ICAM contribute to such a movement of the leukocyte into the tissue. So leukocyte is kind of immune cells. Okay, so immune cells, they keep traveling in the circulation system throughout the body. But once the local tissue need the immune cell, for example, they have been attacked by some pathogen, or they need to trigger certain local immune response, they need to recruit those immune cells. Okay, so you need to those tissue or the blood vessel endothelial cell need to stop those immune cell through the ICAM binding, and they will bind to the blood vessel and then go into the local tissue to trigger the downstream immune response. That will be the process we will introduce here.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. At sites of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G-protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor, and the consequent activation of a subunit of the G-protein. The activation of the G-protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the four steps of a process called leukocyte extravasation.
Okay, so let's use a cartoon to show the whole process. So in the steady state, this is endothelial cell, and it's a kind of traveling leukocyte. So they don't expose to the ICAM, many ICAM or the P-selectin on the surface of the endothelial cell. But once they have been activated or become the inflammation, they need to trigger the downstream immune response. Those endothelial cells will express a PAF, which is a platelet activating factor, and will bind with the PAF receptor on the leukocyte. Okay, and also they will deliver the vesicle containing P-selectin onto the surface. So P-selectin will bind with the carbohydrate on the surface of the leukocyte. Then such a binding will activate the integrin. So integrin will change from the banded conformation to the kind of straight conformation, then become activated. And those activated integrin will bind with the ICAM that is expressed on the surface of the endothelial cell. And finally, they will form super strong interaction and then stop the leukocyte. They don't travel in the circulation anymore. They will stop there and change their shape and finally go into the deeper areas for the following kind of to do their job, to kill the pathogen or to do other immune response. Okay, that will be the leukocyte extravasations. Okay, and also such a kind of, let's just give you an example. The interaction between the cytoskeleton or the membrane component and extracellular matrix is important. If this abnormality happens, it will trigger certain disease. For example, Duchenne muscular dystrophy is DMD. For this patient, you will see they have a strong muscular dystrophy. They have really weak muscles. That is because the researcher found a patient contained the mutant dystrophin. Dystrophin is this protein, okay, here. It will connect with the huge protein complex with the cytoskeleton, okay? And those are protein complex. Normally, you will interact with the laminin, which is one kind of ECM proteins, and stabilize whole things. If you do not have a dystrophin, it will lose their connection and disassemble those protein complex. And such a muscle cell, they will freely kind of move in such a kind of with the ECM, and it triggers many different aberrations, and finally will damage the membrane, and the muscle cell will become really unhealthy. Finally, just trigger muscular dystrophy. That will be the mechanism to trigger DMD. But again, if you have a mutant DMD, now we have many CRISPR-Cas or genome editing tool. We have a chance to kind of really cure such diseases, in the futures. Okay, so here in this section, we just introduce EMT, which is a transition between non-mobile epithelial cell and mobile mesenchymal cells, and in multi-celled integrin will form the adhesion structure. We call that the focal adhesion complex. And also is regulated by the conformation change of the integrin. So there will be two conformations of the integrin, low affinity here and high affinity active form here. And the platelet activation, platelet activate the integrin to form the blood clot, okay? And in the ICAM will be three major CAM will be the new UNCAM, ICAM, and the GEMS. Okay, GEMS, you can find that in the tight junction, and ICAM will be the one involved in the leukocyte atrophisation. We already introduced the whole process here, okay? Okay, so we finished the animal part, then it will come with the plant part shortly, okay? So the plant, this will be the one plant cell, this will be the plasma membrane, the other enabling plant cell. Their extracellular matrix are basically cell wall, and their cell wall is not the protein, it's a polysaccharide, mainly polysaccharide, okay? Actually, those are kind of biofilm, the mainly source of the biofilm people use in the to synthesize that in the factory. Okay, so it will be several layers of the cell wall. The thinner one will be the primary cell wall, and the thicker one will be the secondary cell wall. And it also one kind of area called the middle lamina between the two cell wall here. Okay, and you will see many different sugars in the cell wall: cellular, microfibril, pectin, and hemicellulose. Okay, they will also bind with many different water because some of them will carry the negative charge, and it's also kind of selective filters. Is only allowed the molecule larger than 20 kilodalton cannot really go across the cell wall. Okay, so the animal cell they use NMP metalloprotease to digest this ECM, but for the plant cell, they use a different mechanism to do that, okay? So for the plant cell, for example, in the event called phototropism, okay? So which means the plant will grow toward the sun, okay? Is also kind of regulated by such reactions. So when the sun moves to the one side of the plant, toward that plant, auxin is the hormone of the plant. It's one kind of really important plant hormone. They will accumulate to the other side toward that plant. And the auxin will activate this will be the plant cell, will activate the auxin regulated proton ion channel. It will keep transport proton to the extracellular space and make extracellular space more acidic. And those will kind of help acidic proton will help to loosen those polysaccharide, will loosen the cell wall. Then the cell they will keep uptake keep uptaking the water to extend their cell body, and finally it will make this side of the cell will become larger, and then they will grow toward that some direction. We call that phototropisms. Okay. Between the cell, there will be one spatial structure called the plasmodesmata. It's basically connect the cytosol and the ER lumen between the two plant cells. Okay, so the ER lumen will connect through this structure, and this ER lumen we call the desmoid tubule. Okay, so ER protein can transport here, and the cytosol part will be called the annulus. So the soluble one can transport between these two cells, and the ER lumen protein can transport here. Okay, that will be only found in the plasmodesmata. So basically, this plant cell, they do not have a strong identity. Each cell, they share the same protein, they share the same composition. They do not have a strong identity. Okay, you don't really tell it is an A cell or B cell, but the animal cell, you sometimes can really tell this is one kind of cell and the other kind of cell during development. Okay, okay. Let us go back to the animal cells. There will be one structure called the tunneling nanotube, okay, which was first described about 20 years ago, which is a thin membrane channel. Okay, it's the diameter of the tunneling nanotube, or TNT usually stand. TNT, people use the TNT in short. The diameter about 50 to 200 nanometer. Basically, it can deliver or share everything inside the cell, right? But not the nucleus. Okay, everything. Organelle, it could deliver vesicle, cytosolic component, vesicle, organelle, ER, Golgi, endosome, mitochondria, or even virus particle. Okay, and it can even share the signaling, electrical signal, because the two membrane become the one pool. Okay, and it can even generate some certain mechanical signal. It can pull the other cell or push the other cell. Okay, yeah, this will be the two cell, and it connect with the nanoparticle TNT and to deliver such things. And this will be the 3D structure of those two cells connect with the TNT. Basically, the TNT is a sky bridge. They do not lie on the ground. They actually suspend between the two cells. You will see, right? Okay, and you will see this will be the researcher just labeled the component in this cell, and they found such a TNT and deliver the molecule to the other side. And this will be the mitochondria traveling through those TNT. And such a reaction is triggered regulated by P53 and the EGF receptor signaling. And who are donator, who are recipient. So researchers found that the unhealthy cell will be the one to generate the TNT and release the component to the healthy one. And in their paper, they claim why they need to do this, because it can improve the survival rate of the whole cell population. I don't buy it. It doesn't make sense, right? Who cares? To me, it's like I'm going to die. How can you survive there? So I just release some garbage. Whereas to you, that makes sense to me. Okay, but you need to be positive in the paper. That is understandable. Okay, yeah. Basically, when I read the TNT paper, they don't do good stuff. They are bad guys. They always share. For example, if I had been infected by virus, HIV virus, I just phone the TNT to the neighbors and release the HIV virus to the others. Okay, it's also happened to the cancer cell. The cancer cell on the right side, on the left side, this will be the immune cell. The guy tried to kill the cancer cell, right? They will phone the TNT and try to uptake the functional mitochondria from the immune cells through the TNT. And they will kind of inside the cancer cell, they will kind of modify those mitochondria and become really kind of damaged mitochondria and release back those kind of back mitochondria to the immune cell. So immune cell will become senescent, become really old, become old guys, and ineffective to kill the tumor cell. Okay, basically they are doing really bad thing. They are not good guys for the TNT. I don't see any good meaning about the TNT, okay, so far. Okay, so this will be the image we took in our lab. We labeled the two mitochondria in these two cells. You will see the mitochondria can really move in such a long connection TNT, okay? So this will be the side view, okay? Okay, so in the final section, we just introduced the key component in the plant cell wall. It contains many polysaccharides. It's a cellular microfluidic, it's a pectin and a hemicelluloids, okay? And we also explained how plant cells to kind of remodel their cell wall. They use auxin to activate certain proton pump to degrade the ECM to facilitate the cell migration. They can uptake the water to extend their cell body, okay? And also we introduced about the plasmodesmata. It can connect with the communicate with the ER lumen and the cytosol. The TNT tunneling nanotube can transfer molecule between the animal things for vaccines, okay? That would be kind of the content I introduced.